Good morning, class. So today is geology lecture. We are going to see a new topic that is about the fermentation and its type. So there are more knowledge we are going to take about what is fermentation topic, how we can going to perform the process of fermentation, and what are the major types of fermentation. Let's begin with the aims and objectives. So, if we see what are the major objectives and aims which we are going to fulfill during our lecture. So, first we have to go through with the introduction part of the fermentation. What is fermentation? After that, we are going for the historiological background, like how the fermentation come in possession, type based on respiration. So when we talking about the fermentation, there is a term that respiration will be going to introduce. And on the basis of this respiration, we have to see the aerobic and anaerobic fermentation. Fourth point is going to be in our lecture is a lactic acid fermentation. So on the basis of lactic acid fermentation, we are going to see the homo and heterolactic acid fermentation. After that, we are going through with the mechanism of fermentation by having a pre-definition theoretical aspect about the fermentation. You needed to be see how the fermentation process take place. After that, product of the fermentation. When we go through with the mechanism, we also needed to see what kind of products can be formed by the process of fermentation. Fermentation as a food preservation technique, and then in an application part, like at what or for what things fermentation can be used. In that scenario, we are going to see how we are going to use the fermentation for our food preservation. Advantages and disadvantages of fermentation. So both the benefits and the disadvantages. We will going to see that. And lastly, and very important, the importance of fermentation. So these will be our following part for the entire topic. Let's begin with the very first introduction and overview. So fermentation, as we know, is a metabolic process. So everything which is going inside our body, any anabolic reaction, any catabolic, breaking down of molecules, synthesizing the molecule, all are come under the metabolic process. So the process of fermentation. Fermentation also is a metabolic process. What the basic process that has to be done under the fermentation is that the conversion of sugars to acid, gases, or alcohol. So basically, what we do, what our major goal under the fermentation, that we are going to convert the sugar molecule into acid, into gases, and into alcohol. So these three things can be generated by the process of fermentation. It occurs in yeast, bacteria, and also in oxygen-starved, or you can say the things which do not have the facility of the oxygen, muscles, cells, or in the case of lactic acid formation. So basically, we see what is the location of the fermentation process. So this process can be happen inside the microorganism like yeast and bacteria, and also that can be done by the oxygen starved muscle cell, or if we wanted to see the lactic acid fermentation. It is a type of fermentation, lactic acid fermentation, and that has to be done by in the anaerobic condition, or you can see the oxygen starved condition. Fermentation is a chemical process by which molecules such as glucose are broken down anaerobically. Normally, when we are doing this glucose breakdown process, that happens in an aerobic condition. But if we're going through the process of fermentation, then we have to be do the breakdown process of sugar. That condition, most of the time, that condition should be anaerobically. More broadly, fermentation is a foaming, or you can say foaming means unhealthy, because if we are not using the very vital thing, oxygen, in any kind of process, so that thing can be named as an unhealthy process. So that occurs during the manufacture of wine and beer, a process at least 10,000 year old. So basically, it is another perspective why we are calling them the unhealthy, because they can be used for manufacturing wine and beers. So this is not a healthy thing for our body. They are cancerous. They can make another problem like liver failing, kidney problems. The body have to face a long thing if a person have a habit of drinking. So that's why this wine and beer, they can cause a separate disease things inside the body. So that's why they are calling the unhealthy products. So but this process of fermentation, that is very old. You can see this process is like 10,000-year old process. The frothing result from the evolution of carbon dioxide gases, though this was not recognized until the 17th century. But if we see, like, we know that something was there a very old time, but we don't have a proper evidence of this fermentation process. Is that actually the fermentation process is present at like 10,000 years ago? So this, you can say that when we having a proof, like as I already told you, when we are studying the microbiology historiological part, we know then there was a era when the people was just meaning the thing by seeing them, or you can say they do not need any kind of proof to mind something. But that was not good. We need sometime, we need proof in a scientific era, in a research world. We need proofs to perform something, or if you wanted to believe, you wanted to make believe other people to your talk, you have to give them some proof. So that's why majorly with proof the fermentation process recognized by the 17th century. French chemist and microbiologist Louis Pasteur in the 19th century used the term fermentation in a narrow sense to describe the change brought about by yeast and other microorganisms growing in the absence of air anaerobically. So basically, after the 17th century, when the Leeuwenhoek and other has been described the microorganism, the Louis Pasteur, he was a legend in the case of microbiology, in the case of medical microbiology, vaccination. He was a very important man of the science or history, which make a lot of discoveries, which make things to see possibly with the proof. He was the one, you can see, the father of the major modern techniques today. So basically, he developed or he showed the other people that the this thing, the fermentation, that process, the name even fermentation, that was first given by this Louis Pasteur. So he told everyone about the yeast and other microorganisms that they are growing in the absence of air. So that's why he called them the anaerobic, means without air, a process, a metabolic process can go in there. So that's why this process can be called an anaerobic process. He also recognized that ethyl alcohol and carbon dioxide are not the only product of fermentation. So when he was studying all the process of fermentation, a new discovery has been made that not only alcohol and carbon dioxide, these are just not the two products which can be developed by the fermentation. There are more things, there are more kind of gases, there are more kind of substrate that can be formed when the fermentation process happens. Now, on the basis of all the discoveries, what is the major definition that has been made? That the fermentation is a chemical transformation of organic substances into simpler compound by the action of enzyme, complex, organic catalysts which are produced by microorganisms such as mold, yeast, or bacteria. So you can say that this is a chemical transformation, and chemical transformation of what? That organic substances. And this transformation will be into the simple compound by the action of enzyme. So enzyme will going to be need to break down of these complex thing into the simpler compound. Complex organic catalysts we are going to need, which are produced by the microorganisms. So these things, the enzymes, the complex organic catalysts, they are have to be produced by the microorganism, as we named here some of them, mold, yeast, or bacteria. So they are the unique kind of bacteria which have a very potential to do the fermentation process. Enzyme acts by hydrolysis, a process of breaking down and producing the complex organic molecule to form the smaller. So basically, enzymes, what they are going to do here, they are doing the hydrolysis process. So when an enzyme is doing the hydrolysis process, clearly they are breaking something there. So a process of breaking down pre-digesting complex organic molecule to the simple molecule. So basically, what they are doing, these enzymes eating something or breaking down something to form them from the complex to the simpler compound or the nutrients. You can see the combination of this sugar and yeast, they are going to be collectively form the fermentation process and secreting as a byproduct alcohol and carbon dioxide. Now, take a deep look in the history of fermentation because the process rely on their historical mentors. So basically, this whole fermentation process, it's very, it's very old, and this was a natural process. Man didn't make it. It's naturally present in our environment. So this is a natural process. People applied fermentation to make products such as wine, mint, or fermented honey, water, cheese, beer. So a long things can be made from the fermentation process. Before the biochemical process in the 1850 and 1860s, Louis Pasteur became the first zygomarist. Basically, zygomarist is the one who can study the fermentation process, or you can say who is like the father of the fermentation process. So, the zygomarist, which is a term that is specified for the person who is very expert in the process of fermentation, and we had that person, the Louis Pasteur, who did all the information. He gathered all the information about the fermentation and delivered it to along the world. A scientist who studied fermentation, when he demonstrated fermentation by the cost by living cell. So first, when he talked about that. The whole phenomena that is going to be done under the anaerobic condition, but he also demonstrated the phenomena that has to be done by the living cells. So the first solid evidence of the living nature of yeast appeared between 1337 and 1838. The solid evidence that was given by the Pasteur of the yeast that it is a living thing, and their phenomena was supported by several other scientists and his colleagues, like when three publications appeared by the C. Sagnot, Dalator, T. Swan, and Frank Quittenison. So these were the very famous microbiologists, or you can say the scientists of that time, who basically support that the yeast is a living nature of life. Each one of independently concluded as a result of microscopic investigation that yeast was a living organism that produced by budding. So basically, all the paper they have demonstrated about the yeast, they clearly give the evidence about that living, that yeast is a living thing, and if something is living, they can be reproduced. So basically, their reproduction manner by the budding process. The word yeast should be noted, trace its origin back to the Sanskrit word meaning boiling. So basically, when we see there where it come from, so that was from a Sanskrit word that is meaning is a boiling. So it was perhaps because wine, beer, bread were each basic food in the Europe that most of the early studies on fermentation were done on the yeast, which they were made. Soon bacteria were also discovered. The term was first used in English in the late 1840s, but it did not come into the general use until the 1870s, and then largely in connection with new germs theory of disease. So basically, all this process that was done on the early 19th century, when the Europe and their basic need, their basic food, like in India, our basic food is like wheat, rice, and pulses, cereals. But if we talk about the European countries, they do not have that luxury like us. So basically, they just live on the condition of having bread with them or some bakery products. So bakery was very famous in the European country. Like now in 21st century, we are adapting the things like bread or you can say the beer or wines. But this things would not belong to us. We are just far beyond them those because they are not as healthy as they look. The thing which we eat are the more healthier in comparison to the food nutritional value. So basically, they are eating bacteria and late 1840s it did not come into the general use until 1870. So if we know that the coined term bacteria's fermentation, they were introduced till the 1814 but not have a very much in use by 1870s. The view that fermentation was a process initiated by the living organism soon aroused fierce criticism from the finest chemist of the day, especially Justus von Liebig. So, whenever a scientist made a discovery, whenever someone do something about new, so there are some people who always criticize it. And I'm telling you, criticism or criticizing something that is good, because when we criticize something, if the founder is determined about their foundings, they started to gathering new evidence to make the people believe. So finding this new evidences, finding the new proofs, they make the research more good, developed, or you can say healthy research has to be done only by when you have oppositions, when you have criticism. So then only when you have these things, then only you get more motivated about your finding, and you get gathering extra evidence to support your thing. So that's why the other scientists like Justus von Liebig, Breslin, Frederick Waller, they have a new way of the life, or you can say they do not believe in these things. So this view seemed to give new life to the warning mystical philosophy of vitalism, which they had worked too hard to defeat. Proponent of vitalism held that the function of living organism was due to a vital principle, life force, cheeky prana, distinct from physiochemical forces, that the purpose of the life was not expensible by the laws of physics, chemistry alone. So basically, like these criticism, they made a new discovery to bond. They make it like of this force of living that was come into the consideration until at that time till that time the forces was has been, you can say, the very famous about the physics and the chemistry process, but the alone chemistry and physics process cannot explain the reason behind the life. So this living force, or you can say the vitality, that is a very important term when we talk about the biological sciences. Physics and chemistry, they are the supportive support, but they are not the actual core of the vitality, or they are not the actual reason behind the vitality. So basically, the actual reason that has to be beyond the, or you can say that has to be behind the living organism, that should be vitality. And these discovery has been made by the several criticism of that particular era. So, as well as said soon, the vitalists play a key role in debate on the nature of gradually recognize that yeast was a living organism. Its exact function in fermentation remained a matter of controversy. So basically, now after that, the that was very clear that the yeast is a living organism, but their exact function, like everything have their some functions, some different functions, some complicated functions, some combination function, some have a just one function. So basically the controversy was about that is the fermentation is the exact process that has to be done by the yeast. So the chemist still maintained that fermentation was due to catalytic action and molecular vibrations. So some of the chemists, they do the fermentation, or they see the cause behind fermentation is a catalytic action and molecular vibration. The debate was finally brought to an end by the great French chemist Louis Pasteur. So again, our infamous Louis Pasteur come in a picture, and he ended all the debate that has been, or all the controversy which has been made by the yeast function for the fermentation or not. That during the 1850 and 1860, in a series of classical investigations, proved conclusively that the fermentation was initiated by living organism, In 1857, Pasteur showed the lactic acid fermentation is caused by living organisms. So finally, with the proof, Pasteur showed that the fermentation process can be caused by the living organism, and he showed it to the different scientists, different that time. In 1860, he demonstrated the bacteria cause souring in milk, and a process formerly thought to be merely a chemical change. And his work is identifying the role of microorganisms in food spoilage lead to the process of pasteurization. So basically, as he found it that the sum of the bacteria can do the things with the milk and change their chemical composition. At that time, preventing that thing, he made a new discovery about the pasteurization for long life of the milk. Working to improve the French brewing industry, Pasteur published a famous paper on the fermentation. For the brewing industry revolution, that has to be done by also the Pasteur by uh, publishing a new paper on the fermentation, which was translated into the English as the studies on fermentation. He defined fermentation incorrectly at that time. He write it like the life without air, but correctly showed a specific type of microorganism caused a specific type of the fermentation and specific products. So basically, he gave a wrong name, but his motive was very correct, very evidenceful, because he showed a range of microorganisms that cause specific type of fermentation and specific end products. In 1877, the era of the modern medical bacteriology began when Koch, a German physician, he invented a successful vaccination, and Pasteur showed that the anthrax bacillus caused infectious disease anthrax. The epic discovery led in 1880 the partial general germ theory of infectious disease, which postulated for the first time that each such a disease was caused by a specific microorganism, and Koch also made a very significant discovery of a method for isolating microorganism in pure culture. So basically, they said everyone that our microorganism can cause discovery, and we can cure the discovery by the microorganism. And for proving this point that yes, microorganism can cause diseases, this man, our coach, he cultured the microorganism, and he cultured and injected in some mice and shown that a healthy mouse is going to die just by injecting some bacteria. So that was all about the historiological functions. Now we will see the type of the fermentation. Basically, type based on respiration, if we see, there will be a two type, aerobic and anaerobic. If we talk about aerobic fermentation, that means oxygen should be present. Wine, beer, acetic acid, vinegar, these are they need oxygen basically, and in the primary or the first stage of fermentation. So if we wanted to initiate or make these things, we needed the oxygen in a very primary first stage. When creating acetic vinegar, for example, exposing the surface of the vinegar to as much oxygen as possible, creating a healthy flavorful vinegar with the correct pH. So, if we wanted to make a healthy vinegar with the correct pH, then we needed as much oxygen as we can provide them to be formed. This is a process, anaerobic metabolism fermentation. You can see there is a two type in a yeast cytosol and a muscle cytosol. And you can see the glucose by glycolysis conversion into the pyruvate, and pyruvate dehydrogenate DNP carboxylase. They will be going to convert into the lactic acid and acetaldehyde, which going to be converted into ethanol. So in a yeast that can be make the alcohol formation, and the muscle cytosol they can be going to make the lactic acid formation by the fermentation process. Anaerobic fermentation. So as aerobic need the oxygen, same the opposite in anaerobic, they are not going to need any oxygen for their fermentation process. So in anaerobic fermentation is a method cell used to extract energy from carbohydrates when oxygen or other electron acceptor are not available in the surrounding environment. So basically, when these things which are designed to provide the energy and they are not present there, so at that time, for every process need energy. So they have to find a new source for having the energy, extracting the energy. So they find their new sources by extracting energy from the carbohydrates. Now, this differentiates it from anaerobic respiration, which does not use oxygen but does use electron accepting molecule that come from outside of the cell. The process can follow glycolysis as the next step in the breakdown of glucose, the other sugar, to produce molecules of ATP that create an energy source to the cell. So they are going to use the glycolysis pathway, which is going to produce the molecule of ATPs, and that ATP will be utilizing in this fermentation process. There are two types of the anaerobic fermentation. First is a ethanol based fermentation and second is a lactic acid fermentation. So this are both ethanol fermentation and lactic fermentation. They both come under the anaerobic fermentation. Both restore NAD to allow a cell to continue generating ATP through glycolysis. If you remember all the cycle of metabolic activities, so you know that NAD can be created the ATP. So ethanol fermentation convert two pyruvate molecule and the product of the glycolysis of the two molecule of ethanol and two molecule of carbon dioxide. So this will going to be product by the ethanol fermentation. The reaction is two-step process in which pyruvate is converted to the acetylaldehyde and carbon dioxide first by the enzyme pyruvate decarboxylase. So basically there are two type of the organelles we'll be using here. You can see there lactic dehydrogenase and pyruvate dehydrogenase. After that alcohol dehydrogenase that will going to convert them to ethanol and the carbon dioxide will be releasing the byproduct here. You can see here rather changing from the pyruvic acid to the acetaldehyde that will going to use carbon dioxide the byproduct. Yeast and certain bacteria perform ethanol fermentation when pyruvate from glucose metabolism basically is break down into ethanol and carbon dioxide. The net chemical equation for the production of ethanol from glucose is glucose converted into ethanol plus carbon dioxide. Ethanol fermentation is used in production of beer, wine, and bread. It's worth nothing that fermentation is the presence of high level of bacterium results in the production of small amount of methanol, which is toxic when consumed. So basically, this is the side products that has to be or can be made along with different other fermentation products. You can see alcohol fermentation occur in yeast by the glucose. You can see they are making one from the ADP phosphorylating to the two ADPs converting into the pyruvate by again pyruvate dehydrogenase, converting them to acetylaldehyde. You can see NAD converted into NADH, and this NADH is going to be the source of ATP in a further formation. And after that, they will be converting into the ethanol process. So, lactic acid fermentation is a biological process by which glucose and other six carbon molecule, when we know that glucose is a six carbon molecule, are converted into the cellular energy and metabolic lactate. So, the pyruvate molecule from glucose metabolism may be fermented into the lactic acid. So the lactic acid fermentation is used to convert lactose into the lactic acid in yogurt production. So basically lactic acid fermentation, what they have to do in that phenomena, they are going to convert the lactose into the lactic acid, and in this way the yogurt will be take place after by the milk. It also occurs in animal muscles when the tissue require energy at a faster rate than oxygen can be supplied. So basically this can also be happen if we need a more amount of the energy than oxygen can supply. So at that time also, the next equation for lactic acid production from glucose is glucose will be going to be converted into the lactic acid, and the production of lactic acid from lactose and water may be summarized like: first, the lactose is C12H12O11. That is a formula, molecular formula for the lactose. With addition to the water, they are going to secrete the lactic acid. So you can easily see lactose if they are going to hydrate it, they are going to convert into the lactic acid. So yogurt is made by fermenting milk. It is high in protein, calcium, Probiotics means good bacteria. Here's how they make yogurt and look at the chemistry of yogurt. So this is how you can do the process when the milk is converting to the yogurt formation. This is how the process you have to follow. Now, type of the fermentation, if we see in the lactic fermentation, there are two types of the lactic fermentation. One is a homolactic fermentation. What happened in the case of homolactic fermentation? The fermentation in which only the lactic acid produced. No other byproduct has to be produced when the homolactic fermentation happened. Opposite, heterolactic fermentation. In this fermentation, the lactic acid is produced along with some other gases or major some byproduct can be formed. In a heterolactic condition, homolactic there will be no other byproducts. You can see here diagram of lactic acid fermentation. Glucose through the glycolysis converting to pyruvate, pyruvate lactic acid dehydrogenase converting into the lactate. So this is lactate and going to add up with the water and convert it into the lactic acid. Mechanism, if we see, fermentation takes place when the electron transport chain is unusable, often due to lack of fine electron receptors such as oxygen. So basically electron chain, that is electron receptor chain, that is basically for the producing ATP, but if it is not having the final product or final receptor of the oxygen, they're not going to work out. So become the cell's primary means of ATP production. It turns NADH and pyruvate produced in glycolysis into NAD and an organic molecule, which varies depending on the type of fermentation. So, in the presence of oxygen, NADH and pyruvate are used to generate ATP in respiration. Normally, what happens when the oxygen present, NADH and pyruvate they are going to use to generate ATP in the respiration process. This is called oxidative phosphorylation. So, in the presence of oxygen, when NADH and pyruvate they are going to make the ATP, this process is called the oxidative phosphorylation process, and it generates much more ATP than the glycolysis alone. For that reason, cell generally benefits from avoiding fermentation when oxygen available. So basically, cell do not go under the process of fermentation if the oxygen is available. They just only go under the process of fermentation when the oxygen is not present. The exception being obligate anaerobe, which cannot tolerate oxygen. But if we talk about some anaerobes, microorganism, who cannot tolerate the oxygen, there no choice for them. They have to go through the fermentation process. The first step, glycolysis, is common for all fermentation pathway, either it is aerobic or anaerobic. Here you can see the glucose plus 2 NAD production plus 2 ADP plus 2 phosphate that will be converted into the lactic acid, then another 2 NADH, 2 ATP, 2 water molecule, 2 hydrogen, that pyruvate will be converting into the lactic acid. Now the you can say PI is an organic phosphate. 2 ADP molecule and 2 PI are that is a phosphate molecule are converted into the 2 ATP and 2 water molecules via substrate level phosphorylation. So this whole process, this is called the substrate level phosphorylation, where 2 ATP and the 2 water molecule is going to produce. So 2 molecule of NAD are also reduced to NADH in oxidative phosphorylation. The energy for ATP formation is derived from the electrochemical proton gradient generated across the inner mitochondrial membrane. So this whole energy formation that is also be affected by the electron proton gradient, which had to be developed around the mitochondrial matrix. Via the electron transport chain, glycolysis has substrate level phosphorylation for the ATP generation, and humans have used fermentation to produce food and beverages since the Neolithic age. So this whole process of fermentation, that is not a modern process. That is beyond our thinking. That is a very old process. You can see the Neolithic age. I told you about the geological time scale. So these, if you see that they are the thousands of the year old. Fermentation is used for the preservation in a process that produce lactic acid as found in such sour fruits as pickled cucumbers, kimchi, yogurt, as well as for the producing alcoholic beverages, wine, beer. And fermentation can even occur within the stomach of animals, such as humans. So basically, fermentation process also happen inside our body. We are animals. You can see there how they can be made. Recipe shopping for tourists. No, not needed. Yeast you all have at home. You can just made up with the steam grains inside them. You can boil it for the 16 minutes. Then after you can cool it down. Just mix it and leave it for the weeks. Like primary fermentation will be held in a first week. Secondary fermentation second week. Bottle and condition by the 2 week. You can see the fermentation process have to be on their peak level. Now, the products we can make of the fermentation, that can be wine, beer, lactic acid, vinegar, yogurt, cheese, sauerkraut, kimchi, peppercorns, a lot of soy sauce, everything we can make by the fermentation process. That's why this fermentation process is very old, very much industrial value. Food industry, they are finely used in every step of the food bakeries. You can see how the glucose conversion into the pyruvate by the glycolysis and by this microorganism, how the pyruvate will be going to convert it into these fermented products. Like this are the organism and this is our pyruvate. So by the this Propionobacterium, Aspergillus, Lactobacillus, Streptococcus, Saccharomyces, Clostridium, Echinocobacter, all these things can be converted into the Swiss cheese by the Propionobacterium, and by the Aspergillus lactobacillus streptococcus, lactic acid will be produced and they will be called as cheddar cheese, yogurt, soy sauces. Saccharomyces, they are specifically for the carbon dioxide and ethanol, so they are going to make a wine and beer. Clostridium, acetone isopropanol, that will be held in the nail polish remover, alcohol, ceramic alcohol, lab alcohol that can be used. Escherichia actobacter for the acetic acid production and make the healthy vinegar. Next, you can see here fermentation as a food preservation technique. So, fermented food are foods that have been prepared in a way so that the bacteria naturally found within them starts to ferment. So, the fermentation, also known as a lactofermentation, is a chemical process in which bacteria and other microorganisms break down starch and sugar within the food, possibly making them easier to digest and resulting in a product that is filled with helpful organisms and enzymes. So basically, in this case, we can preserve our food in a longer, like in the pickle, or you can say the sauces. They are basically as a slightly fermentation process happen for storing our food for so long. You can see them; they can be stored in this way. Now, what are the advantages and disadvantages of fermentation? That is very important. Advantages of fermentation, that are lactic acid can be produced and it can produce energy for ATPs. So basically, by the fermentation process, we can be produce the lactic acid, and if we are having the lactic acid, we can produce the more energy or ATPs. Disadvantage of fermentation, that are production can be slow. This is a slow process, not happen in a day or hours. That will take weeks or months. The product is impure and need to have further treatment, and the product carries a high cost and more energy. Basically, this is impure because when we think about something, like some people didn't even eat yogurt just because they have a mindset that there are a lot of bacteria present in yogurt. But yogurt is safe because they are made up of the nice bacteria. So, but still, some people have this in mind, like the bacteria. Even though this is correct for some instant, that these products have to be processed later. Okay, you cannot just eat after the fermented thing because it can be cause not all the good bacteria are there. Some bad bacteria are also there. So you need it to be processed, it, treated it, then only you can use it. So, and this whole process is going to carry a high cost and more energy. So this thing will needed more energy and more cost. Fermentation can provide a rapid burst of ATP in muscle cell. Muscle cell, if they need a ATP and oxygen is less in that time, So by the lactic acid formation, there will be a high ATP production can be happen inside our muscles, even when oxygen is limited supply. Lactate, however, is toxic to cell. Majorly, lactate is a toxic to cell, but if it is breaking down and making ATP at the same time, that it will be safe. But amount of lactate, if it is more in our body, that surely that they are going to damage to our cell. Initially, blood carry or away lactate as it form. Eventually, lactate build up, lowering cell pH and causing muscle to fail. So basically, what happened when the lactate is inside our body in a much more concentration? So they are going to lowering the cell pH. So obviously that are some acidic. So they are going to lowering the cell pH, and if the pH will be lowering, so the mostly the muscle will be in a fatigue condition, unhealthy condition. Oxygen debt occurs, and the liver must reconvert lactate to pyruvate. So as the oxygen come into form, the liver must have to reconvert the lactate to the pyruvate. Otherwise, that is not healthy. Lactate production is not healthy at all. But it is necessary sometime when oxygen is a deficit in the body. But it had not to be present at all the time there. So that's why they will have to be converted back them into the pyruvate, because conversion pyruvate to the ATP formation, that is a healthy process, and that what we need inside the body. Last importance of fermentation. So fermentation is a process important to cell that don't have oxygen or cell don't use oxygen. Now they are very important to those cells which cannot or have not used oxygen. It allows to cell to get 2 ATP gain from one molecule of glucose even without oxygen. So how it is going to benefit? That they are able to produce two energy molecule even without oxygen. That is used. Fermentation take away the end product of glycolysis, so glycolysis can continue. So basically, they take away the end product of the glycolysis, so glycolysis can continue to meet up more pyruvates, because the pyruvate is not there and they need pyruvate to be make out. So that's why they are going again and again, freeing up the electron carriers and so on. So basically, they can just free up the all the electron carriers in that case, and they just made the glycolysis to happen more and over again. Fermentation is important to the baking industry because it is process that yeast used to produce the bubble of carbon dioxide that make the dough rise. So this whole condition that is very important in the baking industry. Fermentation is important in wineries and brewers because yeast use fermentation to produce alcohol. Yeast can produce alcohol, so that's why wines and brewers, they are basically based on the alcohol production, so that's why they can use them. Fermentation is important in muscle because it allows the muscle to keep getting a little energy from glucose even when the oxygen supply can't keep up with the demand. If the sufficient amount of oxygen is not present inside our muscles at that time also, this process, fermentation process, can give us a little energy to survive. That is also important. So this is all about the fermentation process. Still you have any problem, didn't get anything, like you have to, so you can just ask me. These are some references along for the fermentation process you can go through. Thank you. Still you have any problem, just come to me and ask me whatever you need.